a look now at some additional examples. So we've talked about mistakes that happen during DNA replication. We've talked about endogenous biochemical processes like metabolism causing damage. Now let's think about exogenous mutagenic factors, right? Things that are not necessarily part of the food we eat, but we are just exposed to in our environment. So UV radiation is a big one, especially here in Hawaii, when a big part of our life is enjoying the outdoors. So I think all of you are aware that UV radiation is a form of energy coming from sunlight or contained in sunlight that can penetrate our skin and cause damage. But what is the nature of this UV damage? So this figure here just gives you an idea of how deeply UVA and UVB wavelengths can penetrate into our skin. So the outer layer of our skin, the stratum corneum, right, this is the tough, dry, keratinized layer of dead cells. That's our thick outer protection. But then beneath, you have the living cells, and then beneath that, you have the stroma, much deeper connective tissue. And you can see that quite a bit of UV radiation can actually get down to the level of where you have living supportive tissue down below. So the type of damage that UV radiation causes is very specific. UV radiation cross-links adjacent pyrimidines. So here we have a backbone of DNA, and we have two pyrimidines at this particular site here. Right? The UV radiation, the energy comes down, and the radiation basically causes the molecules, the atoms, to vibrate. And remember, in these atoms are bonded together. For example, the carbons in the ring of one and the carbon on the ring of the other, they should normally be separate. They should only be connected to the part of their own ring. But if you vibrate these enough, you run the risk of this bond essentially being broken by these two carbons shaking around. And if these are close together with a neighboring pyrimidine, they may improperly then rebond across, connecting or cross-linking these two bases together. Now, obviously then, this genetic information at the site is messed up, because when anything like an enzyme like RNA polymerase comes along and tries to read this to make a transcript of this, if this were in the middle of a gene, how is it going to interpret this? Right? It's just this one big blocky molecule now. So that's why our bodies produce melanin. So melanin is produced by melanocytes. So in this micrograph on the left, you'll see that you have keratinocyte cells. These are these larger round ones. And you have sprinkled among them these melanocytes. And the melanocytes make melanin. So there are several different types of melanin. This is the chemical structure of eumelanin, which is one of the critical ones that protects us against UV radiation. I like this molecule a lot because I think it's kind of wonderful. It's weird that this molecule kind of looks like this. But the idea is it has many rings. And the way that eumelanin protects us against UV radiation is that if UV radiation comes and hits our body, it is absorbed by this melanin molecule. So the rings, the carbons in the melanin begin to shake around, and that energy is essentially passed through the molecule and is absorbed that way. And eventually then, instead of it being damaging energy that passes on to the deeper parts of our skin, that energy just becomes sort of dissipated as a little bit of heat. So when you actually start tanning, that means the melanocytes are triggered to make more and more melanin, and that melanin is released by the melanocyte and is taken up by the keratinocytes, that are the cells that are being the most exposed. And you can actually see these keratinocytes have taken in the melanin, are actually wearing them sort of on their surface where the UV radiation is coming in. So it's almost like a protective little hat, right? So as more and more of your keratinocytes pick up more and more melanin, that's how we get a nice tan. But the thing to think about here is, how does this melanocyte know to turn on production of melanin? And knows to do so because it's receiving messages from the keratinocytes that are being damaged. So in other words, right, even though we think about tanning as a way to protect ourselves against UV damage, in reality, the fact that you're tanning already is a sign that your keratinocytes are having their genomes damaged. And that's why they're requesting the melanocytes to make more melanin to help them out. In strong sunlight, it's estimated that we can actually suffer from about 100,000 pyrimidine dimers per hour taking place as a result to exposure to sunlight. But the scary thing about this is not per your body. This is 100,000 pyrimidine dimers being cross-linked per cell in your skin. Right? So that is really phenomenal if you think about how much damage just normal UV radiation causes. And that makes you think about how much we depend upon having things to protect and repair our DNA damage, like the BRCA1. So if you think about this then, if tanning is our natural response to UV exposure, because our keratinocytes are beginning to get damaged, and so they're calling for the melanocytes to make more melanin, right? If that's what tanning is, then what is sunburn? Right? Now, I used to think as a kid, right, I think most of us thought, sunburn is when your skin actually cooks, right? That's why it's called sunburn. Right? Like, look at this guy here. Right? So this guy, he decided that instead of going to biology of cancer class, he decided to go to the beach instead. Right? This is what he gets. So what is that? Right? It turns out that when keratinocytes realize that they have way more DNA damage than they can repair, they have to do the right thing. They can't fix themselves, so they undergo apoptosis. Right? So that's when p53 pushes the nuclear button, so the cell undergoes apoptosis. And that is why sunburn leads to things like inflammation and redness, because as these cells are apoptosing, they're releasing all kinds of bits and pieces that then cause inflammation to happen. And that's also why, as we all know, if you're badly sunburned, your skin peels, right? Because those are the layers of cells that have already undergone apoptosis that your body is now experiencing. But the amazing thing about this is when you get sunburn, consider that this represents billions and billions of your cells choosing to die so that you can keep living. Right? This is what I said before, how interesting it is to think that cells actually know, in a way, when they shouldn't try to keep surviving, and they do the right thing and take themselves out, right? So that the whole organism, you, can continue living. But if billions and billions of cells have to do this, you have to ask yourself, does every single affected cell actually go through with it? And all it takes then is a few cells not to apoptose and stick around instead, and they will continue carrying whatever DNA damage they've gotten. Right? So that's why the more sunburn you get, you're just simply increasing the number of cells in your body that did not apoptose properly and are sticking around, potentially transforming. One other way that mutagenic compounds can damage DNA is not necessarily by removing or breaking the DNA, but they can do it by causing adducts to form. So when we talked about cooking food and burning food, one of the byproducts of cooking food are these polycyclic hydrocarbons. Right? So all that nice charred sort of crust on the steak that we like so much, right? It tastes really good, but it's actually filled with all kinds of things like these benzoapyrene types of compounds that have, again, highly reactive oxygen groups on them. So in this case, this big bulky BPDE molecule that came from burning or charring meat on the grill, it reacts with this guanine base that's part of your DNA in a cell. You can see this oxygen reacts with this amine group hanging off here. And now this big BPDE molecule is stuck onto this base in your DNA. If you look at the structure over here, here it is. Right? This is the big bulky BPDE that has now caused an adduct to form, with this base. And as a result, not surprisingly, because it's big and bulky, it messes up the nice spiral alpha helical shape of the DNA, meaning then that perhaps this particular spot of your DNA and your chromosome, enzymes would have a very hard time getting through it. So if this were right in the middle of a coding region of an important gene, maybe then when this gene tries to be transcribed, or when RNA polymerases try to transcribe it, it gets to this point, and then you cannot transcribe the rest of the gene. Okay, so when we talk about all these types of damages, it sounds kind of scary, but you realize that the majority of the cells that we're referring to in your body that are exposed to these types of damage from things like burnt food or UV radiation, these are very much your fully differentiated cells, right? Either the fully differentiated keratinocytes that are part of the outer layers of your skin, right? They're getting the ones that are being hit the hardest by UV radiation, or let's say the epithelial cells that line your intestines. So those are the cells that would get hit or exposed the most to things like the burnt compounds from grilling food. So that's why, again, we said highly differentiated cells, they're designed to not replicate too much more, right? Because we expect them to get a lot of damage, but it's okay, because if they're not going to replicate anymore and they're just going to stick around for a few days, maybe a week before they are lost, then that's all right. But stem cells then, the ones that constantly give rise to replenishing those differentiated cells, it is a lot scarier to think what happens if a stem cell actually gets damaged in this way. So one of the ways that our bodies protect our stem cells is by physically positioning them deeper away from the parts of our body where there's all kinds of exposure to exogenous factors. So here, for example, you can think of this as being layers of tissue. This area in blue here might be deeper in your underlying tissue. And as you move to the right of the slide, you're getting closer to the surface of a tissue. So for example, let's pretend that this white spot on the slide on the right is actually the lumen of your small intestines or something like that, right? Where you'd have all the food with all the different compounds, natural and synthetic carcinogens, right? So these are the highly differentiated cells here in red. They're the ones that are going to take the hit the most. So the stem cells then are deeper away from that. And importantly, this is a nice slide for illustrating this concept that even though stem cells stick around for a long time and continuously replicate, they're not doing it very often. So the same stem cell here, when it divides, right, one of its daughter cells becomes this green cell here. The other daughter cell basically sticks around and is still the stem cell. This one green daughter cell then becomes part of a transit amplifying cell chain. So in other words, this is the cell that begins to further differentiate, and actually through several rounds of differentiation, you make many highly differentiated cells just from that one cell division of that stem cell back here. Right? And again, these then, the highly differentiated cells, they don't stick around very long. They naturally are lost quickly, or they undergo apoptosis. Right? And so the stem cell then can keep dividing over time, et cetera, et cetera. But as you can tell, it's not very often, because one division can lead to many, many differentiated cells being made. Okay. So other ways that stem cells protect themselves against mutations. Number one, we said they're physically located away from mutagens in your body, and they infrequently divide. So in other words, their job throughout your life is to keep dividing, but it's not something that happens like every day. Also, stem cells are primed to be very sensitive to undergo apoptosis if their DNA gets damaged. So unlike a differentiated cell that might sort of pause its cell cycle and try to fix itself with P53, the stem cell may do that as well, but it won't really try as hard to fix itself. It'll more likely just undergo apoptosis, sort of as a conservative way of trying to just avoid the potential for a problem being passed on to all of its daughter cells. This is also why we talked about the fact that as we age, our skin gets less and less firm and less healthy, right? Because even though our stem cells throughout our life are supposed to be replenishing it, through our life, more and more of our stem cells end up undergoing apoptosis. And so you have fewer stem cells that are left over to replenish your keratinocytes when you're 70 years old. And importantly, those stem cells that are still there in your skin carry some baggage with them over many decades of collecting mutations, right? Maybe not bad enough mutations to cause them to apoptose, but certainly mutations that can pile up over time and mean that the quality of keratinocytes that they make when you're 70 years old is not as good as the skin cells you made when you were 18. Also, stem cells actively pump out small molecules such as toxins. On their surface, there are certain membrane pumps. These are proteins like MDR2, whose job it is to constantly take exogenous or xenobiotic molecules from outside of the cell that have somehow entered the cell and just pump them back out of the cell. And so this is especially true for toxins. So stem cells have a lot of these mechanisms for trying to really preserve their genome. So it should be noted that stem cells, because of this problem, or I should say that by having lots of pumps and pumping out toxins, this is another example of a clever mutation that cancers can actually develop or stumble across. Because you can imagine if a cancer cell stumbles across a mutation that really upregulates the amount of these membrane pumps it has on its surface, then that particular cancer cell might be more resistant to different types of chemotherapies that a patient is being given. And then the last one of these I put in blue because it's kind of a controversial one. And there is this idea of asymmetrical allotment of DNA to daughter cells, meaning that every time your DNA in your cell makes a copy of itself, you'll remember, right, that from one double-stranded DNA, you're going to get two double-stranded DNAs. Right? So in other words, 4 strands of DNA that make up 2 complete alpha helices. But in each one of those new copy double helices, one of the strands is actually from the original template. Right? So there's this idea that as daughter cells differentiate, one of the cells, the stem cell, actually holds on to the original DNA and allows the copied DNA to pass on to the daughter cell. And in this way, it sort of is an attempt evolutionarily to minimize the amount of mutations that happen during replication that actually get passed on to the daughter cell. So I say this is controversial because I think scientists still haven't really determined if this is true for how our stem cells function, or if this is just sort of something that has been seen from time to time in experiments, but we don't know if that's really an important part of how our stem cells really do their job.